in general, I'll start by saying that in general, I'm interested in the interplay between single cells and the multicellular structures that they form. And questions involving, for example, how can we design local rules for interactions to lead to self-organization of specific structures or specific function that we're interested in, and also the reverse type of questions. So how can we identify the identities and interactions between single cells from these kind of global structures or from their features? And today I'll be talking about a specific aspect of this question that has to do with the inference of a tissue structure from, excuse me, from single cell data. So we know that today it's relatively easy to know a lot about the identities of single cell, but to really understand their collective behavior and tissue level or organism level design principles, we need to also know something about their spatial context. And if we do that, then we can also understand better division of labor and patterns of cellular communication and patterns of gene expression across the tissues. So specifically, our motivation is twofold. The first is to infer cellular locations within a tissue, and the second is to infer gene expression. And these two are obviously related, but they're not identical. We can do the second one, for example, without having very good, precise information for the first. So maybe I'll just mention here, I didn't say that at the beginning, I'd be really happy to get questions and comments, or if anything is not fully understood, just let me know as we go. Okay, so the setup that we should have in mind for this talk is that we have cells in a tissue, which we'd like to sequence to know something about their identity. But unfortunately, to do so, the tissue has to be generally dissociated. So we lose any spatial context for the single cells. But now we can do single cell RNA sequencing and get an expression profile for each individual cell. That would be our input. And then from that, we'd like to infer either the cell's location or the gene expression patterns that they confer. Okay, so what is the usual approach of thinking about this type of problem? So the usual approach would be to say that we have some additional type of information. So say a reference atlas. So now we have both single cell data, but also a reference atlas, which is usually an imaging data set that tells us something about the expression levels of a subset of genes for different locations across the tissue. And these may be either at single cell resolution or at lower resolution. And then if we have that, then for each individual cell, we can compare the expression levels of that relevant subset of genes to each of the locations in the tissue that we have that additional information for. And if everything is nice and reproducible, then we can possibly match the single cells to the proper location on the tissue. And there have been a few works that approach that problem from that direction. The problem, and this is where our main motivation comes from, is that we don't always have such a reference atlas. We don't always have we don't always have it existed, or it's not always easy to construct. And also, even if we have it, it may be insufficient, either in terms of the number of genes that it contains, or the spatial resolution of the atlas, or the quality. And then we have a problem and we can't properly match the single cells to locations. So what can we do, right? And I'll mention here that, of course, there have been several efforts to approach this problem experimentally, some done here at the Broad. The problem is that we can't fully rely on them yet, because either they are still technically challenging or at low spatial resolution, or just in general not as widely applicable as single cell sequencing. So we'd like to use the information we currently have. Okay, so if we don't have a reference atlas, then what can we do, right? So we have our single cells, and we'd like to map them onto the tissue, find a configuration of the single cells on the tissue in a way that makes sense, right? So we can think of it, maybe a useful way to think about it is like a puzzle, where we're trying to find, out of all possible configurations, we're trying to find the configuration of pieces, or single cells in this case. So the configuration of pieces in space that makes the most biological sense, right? So that raises the obvious question, what does it mean to have biological sense in this context? How can we rank different configurations relative to others? How can we say that one is more biologically meaningful than the other? So you're very quiet, so maybe I'll ask you what you think. Anyone has any suggestions for how to do that? Or it's too early, maybe? Yeah. Okay. Okay, good. So the first thing is probably the most natural thing, and that's also what we went for. So you mentioned the smoothness assumption, right? Good. So we can think of a few possible hypotheses for how cells would tend to be organized in space. And need for many canonical tissues, when we have, for example, gradients of oxygen or nutrients or morphogenes across a tissue, or we have cell-to-cell communication, or we have some trajectory of cell maturation, or any subset of these, then maybe we could expect to have some notion of smoothness across the tissue. Which would mean that cells that lie close to each other in physical space on top of a tissue would also tend, on average, to lie close to each other in expression space.
But only for some genes.
Yes, so that's my next question. So this is a really important point to highlight, that here I'm not talking about each individual gene, right? This is a property averaged over the entire transcriptome of single cells. So it doesn't mean that we can't have individual genes which don't follow this assumption, because we know we always do, right? It doesn't mean that we don't have certain genes that show sharp spatial boundaries across the tissues, or even salt and pepper patterns. But it does mean, or maybe we can hope, that there are enough genes that would show some signal for this type of smoothness, especially compared to random or shuffled configuration. And hopefully we can pick up on that signal. OK. So let's think about it a little bit more concretely. Say we take this assumption, which we can actually test, and I'll talk more about it later. I'll show a few examples for real data, and that, in fact, this assumption holds there. But for now, say that we take this assumption as our starting point. How can we actually use that to assign the cells to specific locations across the tissue? OK, so as I said, our initial input is cells in high-dimensional expression space, right? Because each cell has an expression profile associated with it, so we can think of it effectively as a point in high-dimensional gene expression space, which many times we saw before that the cells actually are not just randomly spread out in space, but actually lie on some low-dimensional structure, or maybe a manifold, in that high-dimensional space, and that would be useful in a minute. That's one type of information. The other type of information that we have is maybe something that we know about the tissue. So that's not required, but that would be helpful for us. So say we know that the initial shape the cells were generated from. So we don't know where cells came from, but we know that they were taken out of an embryo, and we know something about its shape, right? In special cases, in very well-studied organisms like the Drosophila embryo, where at certain early stages of development, we know exactly where cells are located, not their identity, but that they're equispaced across the surface of the embryo. Usually, we don't have that kind of information, or we don't know too much about local densities of cells across the tissue. In that case, we can construct a grid across tissue and basically transform our problem to assigning the single cells to specific points on this grid. So the assumption from before, which we'll call the structural correspondence assumption, that basically means that we would like the distances between cells in expression space to correspond to the distances between cells in physical space. So that cells that are close to each other in expression space would also be close to each other in physical space, vice versa, and also for longer distances. And we can adjust these later. So we can take direct measurements across the space, a Euclidean distance between cells, or something that has to do with the correlation between the expression profiles. The problem is that if we do that directly, then that doesn't account for the nonlinear structure that the cells actually lie on in high-dimensional space. And we can overcome that by thinking about graphs. So these would hopefully capture better the nonlinear. So if the cells here, for example, lie on a nonlinear low-dimensional manifold, then if we take direct Euclidean distances, let me just give the usual example for that. That would hopefully make it a bit more intuitive. If the low-dimensional manifold looks something like this in high-dimensional space, and we have cells spread across it, then if we take direct Euclidean distances, then these two cells would appear to be close to each other. Although if we want to capture something about the manifold structure, we would actually need to take into account the distances along the manifold. And one maybe easy way to do that is to actually construct a graph. So here, for example, we can say that we can connect each cell to its two nearest neighbors. And then if our distances are actually the shortest paths along this graph, then the distance between this cell and this cell would actually go through all of these and would reflect the underlying structure of the manifold. So again, please ask questions. Yes. Yeah, so here in terms of constructing graph.
In terms of a metric on.
Yeah, so here I'm starting with, for the expression space, for the physical space, I'm starting with Euclidean distances. For the expression space, I'm starting with correlation-based distances, and I'm constructing a k-nearest neighbor graph based on something. Sometimes I know something about the underlying dimensionality of the data, especially for the physical, for the physical space. Even if I don't, there are different ways to approximate it. I also tried a few other things. You could do more sophisticated things. You could use diffusion maps. You can use other measures for that. It tends, at least for the types of data that I try, to not make that much of a difference. So yeah, but that's the, yes?
So this expression space doesn't have any prior knowledge about the relevance of these genes, right? Basically,
the expression space doesn't have?
Any prior assumption about the genes which are like most relevant
to the spatial configuration.
You're weighing every gene equally, I guess, right?
When constructing the graph?
Yeah.
So yeah, so here as well you have some freedom, right? You can choose the most variable genes in different ways. But in general, here I don't assume that I know that I have any prior knowledge about which genes are important for the spatial configuration. And that's actually one of the challenges, to using this framework or similar types of ideas actually also go back and infer that information, because that's sometimes actually the most important thing that you'd like to find out. Yeah, but we don't know that ahead of time. Any other questions? No, okay. Right, so once we have these two graphs, we can compute the distances based on shortest paths along them. Again, we can do it in different ways. And then we end up with two distance matrices. One expressing the pairwise distances between cells in expression space, and one expressing pairwise distances between cells in physical space. And the structural correspondence assumption that I talked about before, what it basically means here is that we'd like to find the right assignment of cells to physical locations in a way that if we shuffle these two matrices correctly, then they would match to each other, right? Ideally, which is not the case because we have different types of scaling and it's not, life is not perfect in general, then ideally we would have exactly the same matrix for both physical and expression space, but only, but in a way shuffled, so that we're not sure. Yeah, maybe let me continue that line of thought, and then if it needs more clarification, then I'll get back to that. Okay, so if we'd like to match these two matrices, this would become clearer when I continue on to the math part, hopefully. Okay. So when we try to compare these two matrices, we would also like to fulfill two other requirements. First, we'd like the assignment of cells to locations to be probabilistic, meaning that cells would not be assigned a single location across the tissue, but some probability distribution over all possible locations. And I'll say a few more words about that in the next slide, why we would want that. But in general, it would be more robust, and it turns out justified both biologically and computationally. And the other thing we would want is that, I said that so far we assume that we don't have any other type of prior knowledge, any other type of reference atlas, right? But if we do have it, or we do have some partial information about it, we'd like to use it, right? To inform our assignment, make sure that our assignment actually matches the information we already have. And then if we can do all of this, then hopefully we can do what we initially set out to do, which is to find the right assignment for single cells and also find the expression patterns over the tissue. Right, so why would we actually like probabilistic embedding as opposed to deterministic one? So there are a few different reasons. The first one is that inherently, this type of embedding between cells and locations is not a one-to-one matching problem. We actually don't expect these two matrices to be exactly the same. And that has several different reasons. One is that even if we knew how many cells were originally in the tissue, even if we knew how to construct the grid perfectly so that each grid point initially had exactly one cell sitting there, then still when we use single-cell RNA sequencing, we know that we don't get 100% of the cells, right? And even if we were talking about reproducible tissues, reproducible organisms, like, for example, the Drosophila embryo at early stages of development, which is highly reproducible, still, even if we sequence many of these, embryos, we would still be expected to be missing some cells, some cellular locations, and on the other hand, sequencing several cells for others, right? So it's not a one-to-one matching problem. So in that sense, maybe it doesn't make too much sense to think about it as a deterministic one-to-one embedding. The second point is that thinking about it probabilistically would make the embedding more robust. And we know that with the type of data we have with single-cell sequencing data, it's noisy and messy for several good reasons. But we'd like to overcome that in our approach. One way to think about it is that if we don't have too much information, then we wouldn't want to get stuck on biased, random, deterministic embedding, but we would like to say that maybe the cell was originated from that area of the embryo and not push ourselves to say just one single point, because maybe we just don't know. Maybe it's just not in the data. There's not enough information in the data. The third point is that it's more efficient computationally, and I'll talk about it more later on. And the fourth is a different kind of point, and that's actually that many times we would be interested in reconstructing stereotypical tissues and not necessarily just one specific tissue that the cells originated from. So that would be relevant for, again, nice reproducible systems. And I'll show that in the type of real data that we can currently deal with. OK, so now that we maybe have the general idea, we can ask, how do we actually do that? How can we map these ideas onto something we can actually solve? And we'd like an efficient algorithm for it eventually. And there was a question. Yeah.
So I was under the impression that often in single-cell data sets, proximity and expression space corresponds more to cell type than to location.
So that's a very good point. And I kind of swept that under the rug and was planning to get to that later. Currently, I'm talking about a specific cell type. So if we have single-cell data, we can first, say at least in the naive situation, it becomes more complicated, and we can deal with that too. But let's think of the simplest one. We can cluster the cells into different cell types, and then take different cell types one at a time. We can later compose these, but you're right. Yeah. Any other questions? OK. So, right. So it turns out that we can actually formulate these ideas as an optimization problem. And let's see what we'd actually like to optimize. So because we're not thinking about it as vanilla dimensionality reduction, we're actually thinking about matching of discrete set of points, the single cells, to another set of discrete points, the locations. Then what we're looking for is a matrix of positive values that takes us from n single cells to m locations. Basically tells us the probability for each single cell in each one of the rows. What is the probability over the entire, to be locally or spatially distributed over the entire tissue, right? So what is that cell's probability to be located in the first bin, the first point of the grid, the second point, and so forth? Yeah.
I'm afraid it's going back to where you were before.
Yes.
And it's because I'm not in the field, maybe, this question. So
yeah, sure.
In terms of the sampling of the single cells from a particular tissue, and so how confident are there bounds on how confident the sampling, how sparse or not the sampling would have to be from the embedded manifold in terms of the 3D? So if you do it to a particular tissue, even if it is one so-called cell type,
yeah.
What's the proportion? Presumably below a certain number, you're not sampling in
Right.
Even if you know the shape of it, the graph would be very misleading.
Right. So what happens usually, and that is actually true for all of the examples that I will present, the 3 examples I will present, and the 4th one that appears in our paper but not here. For all of these, it's not like we're taking just one single tissue and then sample single cells from it, but it's actually, since that part of the tissue is reproducible, then we can sample, or not me personally because I'm not doing the experiments, but people who know how to do that can actually sample many such tissues to hopefully get a good cover for that, yeah. Okay, so we're looking for this coupling matrix between single cells and locations. The first thing that we'd like it to uphold, that's actually the constraint on the matrix in the optimization problem, is that we like it to be consistent with the marginal distribution over single cells and over the spatial locations that we have. If we don't know anything about these distributions, we don't have any prior knowledge about these, we can just set it to a uniform distribution. But this type of formulation actually lets us use additional information we might have. For example, that even if we have tissue, we construct a grid over it, and we're trying to assign single cells to these locations, we may know for certain cases that this location is more densely populated with cells based on imaging data or some other type of information we may have from other biological experiments. Or from the single cell side, we may know that, after some analysis, we may conclude that some cells are of higher quality than others, and therefore we'd like them to dominate the expression patterns that we eventually see, and the lower quality cells maybe to contribute less than the higher quality cells, right? So that gives us an additional knob to play with. But the interesting part is actually the one that follows. And that is how can we actually formulate this optimization over T? What would we like to optimize? So the first term would have to do with the structural correspondence assumption. So here,
Can you write the equation on the board, maybe?
yeah, maybe it would be easier, right?
You don't have to erase it.
Okay. Okay. So the T we are looking for, which fulfills certain constraints, which we talked about. This would be the one that minimizes several different terms. So the first one, maybe I'll start like this. So for the structural correspondence assumption, that basically means that if we have cells I and cells K, right? So this would be a quadratic term. So if we have cell I and cell K that are embedded correspondingly to location J and location L, then we'd like the distances between I and K in expression space to match those between J and L in physical space, right? So a way to do that would be to say that we'd like to minimize some loss function between the pairwise distances between I and K in expression space and the pairwise distances between J and L in physical space. Right. The next thing that we would like to do is to take into account any other information we may have, such as a reference atlas, right? So if we do have a reference atlas, then we can add, and this goes over all possible cells and locations. If we do have a reference atlas, then we'd like to also minimize for each cell, I, assigned to location J. We would like to minimize the discrepancy according to the atlas. Let me just write atlas here. Between the set of genes we have information about between cell I and actually location J that was assigned to. And we can interpolate between these two terms. So that if we have a more extensive reference atlas that we trust more, maybe we would like to weigh it more relative to comparing the geometries in expression in physical space. And finally, we'd like to also push this assignment to be probabilistic. And turns out it also really helps us computationally. So we can also add an entropy term that basically pushes the assignment to say that for each cell, it doesn't only assign to one location, but tries to spread across the tissue. But of course, we have these terms that constrain us from just having a uniform distribution over the entire tissue. OK. Yeah. Oh, yeah. Yes. Oh, so yeah, let me just write it, what it means. So no, it's a matrix. So here it would mean going over all cells, all locations. Yeah. OK, so for each single cell, you can, yeah. So it's basically the entropy over the entire matching matrix. Is that OK? OK. Right. The nice thing for us is that we can actually show an algorithm that solves this optimization problem in an efficient way, actually by using projected gradient descent, where each step turns out to can be reduced to a linear program. Basically, only an adjusted form of the entropically regularized optimal part, which we can solve very efficiently. And yeah, let me just say one more sentence, and then I just want to say that this optimization problem, the way it's formulated here, actually falls very nicely into the generalized framework of optimal transport, where we try to push one distribution, in this case of single cells, into different distribution, in this case of the locations. And we have many good results that we can actually build on to do this efficiently. Yeah? No,
so do you have any assumption that the number of locations should be less than the number of cells?
No, so they can be either more or less. Yeah, yeah. And because it's probabilistic, then it doesn't matter in general.
How do you tackle with the computational complexity of this? Because it's going to be, so you have, if you calculate this entropy, if you try.
Yeah, so it's right. So it's not as direct because it's a nonlinear program. So you actually do it iteratively, and each step actually turns out to be just a process of matrix scaling, which is very efficient. And I can maybe talk to you more about it at the break and during the break. Yeah.
This looks very much like a quadratic assignment problem.
Yeah. Right. Right. So yeah. So actually, when I started thinking about this problem, I thought about it as a quadratic assignment problem, right? Then thinking about also the fact that we would like to match the reference atlas, it became a quadratic plus a linear matching problem. But it turns out that it's actually really hard to solve, right? If you don't have the entropy term, then you don't really have an efficient way of doing that. And using different heuristics, you can get stuck at really bad solutions. And I know because I tried it and it's also very slow, right? But yeah, but it turns out, and again, we can talk about more over the break, if you add the entropy, actually, it reduces this whole problem to something that you can solve in a computationally efficient way, which actually makes it possible to even approach the kind of problems and the kind of data sets that we actually have. Yeah.
Can you use this atlas loss term as a way of choosing good expression embeddings? Do you find the solution here without the atlas loss and then just calculate what is the atlas loss for a certain set of genes or a certain embedding?
I could, but then why do you think it would be better to count for it in the first place?
The embeddings right now are being chosen for possibly other reasons unrelated to doing this reconstruction.
So I'm not sure I got this point. You're saying to first ignore the atlas, reconstruct without it, and then compare to what we have with the atlas, right? So that's actually what we did for the data sets that I will show you very quickly. That's what we did there. So to basically evaluate our own performance, we ignored the atlas, reconstructed without it, and then compared to what we actually know about the tissue, which is basically an imaging atlas, right? So we could do that, but as we add more genes from the atlas that would inform this embedding, it actually improves. And I'll show that also for the third data set. Yeah. OK. So now that we have this approach, which we termed novoSpaRc, it's on the bioarchive, and also we have, it's now public on GitHub, we can actually use it on real data, try to reconstruct real tissues. And I'll go over three relatively quick examples. And the first one is the intestinal epithelium. So again, all of these were, we actually based our, so we took existing data sets. We didn't do the experiments ourselves for this work. So the data set for the intestinal epithelium was taken from two papers published in 2017, 2018. The second one is from the Itzkovich lab, which will also appear in our next data set. And actually, in the intestinal epithelium, it is composed of repeating units. As we said, that's what we basically, that helps us make this reconstruction much more robust. So it's repeating units of crypt to villi axis, where cells originate in the stem cell niche, the bottom of the crypt, and migrate across the villi till they eventually shed off after a few days. So here we have a trajectory of cell maturation, as well as different gradients of oxygen and nutrients. So we thought this would be a good candidate to try out our approach. And first, we wanted to make sure that our structural correspondence assumption holds, right? Because again, we have the single cell data set, but here we also, from the existing data sets, we also had a reference atlas. Using that, it was possible to reconstruct the intestinal epithelium, the crypt-to-villi axis, and then confirm that with other fish measurements. So we could take this data and basically see that, indeed, as we expected, pairwise distances between cells in expression space here on the y-axis increase monotonically on average with pairwise distances in physical space between the different locations across the villi. And now if we use our approach, if we use novoSpaRc to actually reconstruct the locations, we see that it does a pretty good job. So here you see a heat map where the cells originate from either the crypt or one of 6 zones across the villus. And then on the y-axis, we see our predictions for the assignments to different zones. So, and we see that our predictions concentrate relatively nicely along the actual original zones that the cells were originated from. And this works not only on average across all of the cells, but we can also look, across all of the genes, all of the cells. We can also look at individual genes. Here are expression patterns, the original one on the top and the embedded predicted one on the bottom. And we see here spatial specialization for gene groups that are responsible, in this case, for the absorption and transportation of different nutrients across the villi. Okay, the second example would be that of the liver, which is also composed of repeating units called liver lobules, where we have blood flow, which generates gradients of oxygen and nutrients, and we also have gradients of morphogenes, in this case, in the reverse direction. So we use data from a different paper coming again from the Itzkevich lab for the liver lobules. Here again, we see that our structural correspondence assumption holds, so the geometry is corresponding for the cells in expression in physical space. We can again reconstruct the original locations of the cells without any reference atlas. I forgot to say that we did that before for the intestinal, also without any reference atlas, only based on a single cell data.
So this is only considering the hepatocytes. You've taken out like other.
Yes.
Okay.
Thank you. Yeah. So this is looking at the hepatocytes. Yeah.
So again, because basically this is an interesting example for the spatial correspondence assumption, where you would really expect the hepatocytes to have that property, but like what would happen when you have these like rings of cells of the same type that are embedded in there? Like I would actually imagine like the artery, venule, and bile duct cells would actually be more similar to each other than to other things.
Yeah, so as I said before, so far we only focused at each stage at one cell type, here, for example, the hepatocytes, right, where we would expect to see some kind of continuity due to these kind of gradients, right? But definitely one of the next steps is to extend that to different cell types and see how much they vary in space. What can we say about intersecting domains between different cell types and so forth. And we have some initial results, but
I guess they would actually break the spatial correspondence assumption.
Yeah.
Especially because you have like rings of cells embedded within other structures.
Right. It would definitely not be as straightforward as just using this for a single cell type. I agree. Okay. So again, we can look at individual genes. On the left, we can see the original assignment to different lobal layers across the lobal axis. On the right, we can see our prediction. And we see that we can basically capture zonated patterns of expression across and also non-monotonic expression patterns across the lobal axis. Yeah?
The axis that you have is, what's the color? What's the axis? So the other one I noticed was 0 to 1, and this is 0 to 0.6 on the previous slide. The one on the crypt was 0 to 1, and this is 0 to 0.6. Is that what you, so therefore
so this is actually basically taking, so this is averaged, right? So this is taking all of the cells actually originating from the first lobal layer and asking how do they distribute on average across the different layers. So
what's a perfect result look like?
A perfect result would look like just a diagonal,
Or what, would it be 1, not 0.6?
Oh, yeah.
Okay, so I mean, it's probably an amazing result, I'm sure, but this is not as good as the previous result.
So it's a good question because we actually can, by construction, currently, we actually can only compare ourselves to the previous result using the reference atlas. So by definition, it can only be as good, but not better than that. But one of the things that we're working on is doing it for understudied tissues where we can later go on and do the experiments ourselves, and not myself, but along with collaborators, and actually confirm different, Right, and these are a few examples for a few genes that seem to correspond nicely to the original data. But I wanted, because we have only a few minutes, I wanted to go over really quickly some results for the Drosophila embryo, which I think may be most exciting. And we're looking at late stage 5 of the Drosophila embryo development. Because this is a more complicated case, we actually started from taking an imaging data set, the Berkeley data set, which we could effectively dissociate and try to reconstruct. So there, again, we see the structural correspondence assumption holds, as we would expect. And if you look at the top, then you see that the quality of reconstruction increases with the number of marker genes that we use for the atlas, right? The interesting thing is that it saturates and we get near perfect reconstruction when using only 4 or 5 genes. That's our approach is the top line. The bottom line is actually using only a reference atlas, the usual way of using only a reference atlas. And then when we look at specific genes, then we can compare the fish data, the top, to our reconstruction using 2 genes in the atlas, a reference atlas composed of just two genes, or 4. And we see that for 2, we already get pretty good results, and for 4, it seems nearly perfect. But the surprising thing is that we can even do pretty well without any reference atlas, without any genes, right? In that case, of course, we don't have any way to anchor the reconstruction. So in this case, as you see, it's flipped. But it's interesting that it can still capture some information, and I'll show in the next slide something else about that. And I just want to mention that we can also capture the original location of single cells in this case, right? So here we have the location of original cells, for example, original cells on the top, and then our prediction for their embedding at the bottom with 4 marker genes in the atlas. And then when we don't use any marker genes, then first of all, it's flipped, which is to be expected. And also, it's spread out, as again we would expect, because we have less information, so that we wouldn't want to reduce ourselves to just one location if we're not sure about it. I'll skip that. And just say that if we use single cell data, in this case taken from the Rayevsky lab, for the Drosophila embryo, then we can see that we can capture the main spatial domains in the embryo without using any reference atlas. So here are a few genes in rows, fish images on the left, and our predictions for their corresponding expression patterns on the right, which you can see that even for this large and noisy data set works relatively well. Okay, so we're currently working on using these ideas to infer different types of biological processes, different types of structures in single cell data, identifying spatial informative genes, as I was asked before, and also applying these ideas to less explored tissues and generalize them to also disordered tissues where we have different cell types, where we don't have the usual smoothness assumptions we can rely on, but maybe something more complicated we should adjust for. And I'll take 1 minute just to say that, because I think this is a really good audience to interact with, I just wanted to mention a few other things we've been thinking about and working on in the past few years. And I'd be really happy to talk about them offline. I'm at Harvard, so not that far. So the first one is revealing latent structure in single cell data. The talk today was one example of that. We're thinking about other types of such structure more generally. The second direction has to do with thinking of efficient ways to use single cell embeddings or integrate data from different modalities taken from different biological experiments for the same system, or using spatial data or low resolution data to deconvolve it over single cells, all these kind of things is also something that we're interested in. Inference of network structure in general, both for cell-to-cell interactions and gene-to-gene interactions for gene regulatory networks. And finally, analyzing modeling and design of the self-organization, the dynamic self-organization of multicellular system and connecting that to actual experimental systems, for example, organoids and such. So Justus, who's a great postdoc who's working with me, is also here today. Maybe you can just raise your hand so that people know. So I guess both of us would be happy to talk about these more over the break and offline. And I'd like to thank my collaborators, and thank you. Yep.